In our unit so far, what we've talked about is just some basic chemistry in terms of electrons, structure of the atom, a little bit about pH. So now what we are going to be doing is talking about various, we call biomolecules. Okay? Molecules that are important to living things. They are all organic compounds, meaning they contain what? Carbon and hydrogen. Not that they contain all of those elements, but that they contain at least carbon and hydrogen. Some contain all of those elements, some do not. And when we look at the organic biomolecules, sort of group them into categories. And we'll talk about each in turn. So first, carbohydrates we'll start with. And we'll talk about those today and maybe tomorrow. And carbohydrates include polysaccharides and disaccharides and monosaccharides. And we'll talk about some examples of each of those and important types of those carbohydrates. Then we talk about lipids, proteins, nucleic acids, and then we'll talk about some of these other things later on. Okay? But these molecules, many of them are polymers. They are large molecules made up of sort of repeating subunits. And so you're going to have to know the use of these different molecules in living things, be able to identify some of them based on their structural formulas and so forth. So that's sort of where we're going. Great. All right, so start with carbohydrates. So carbohydrates we're familiar with. Their functions inside of living things, they are a source of energy. In fact, they are the main source of energy for living things. Carbohydrates contain about 4 calories of energy per gram. And what is the most important carbohydrate that is needed for energy in cells?
Polysaccharide.
No, not a polysaccharide. A type of sugar, a specific sugar though.
Glucose.
Glucose. Because it's used during what process? Cellular respiration, where the energy from that glucose molecule is extracted and transformed into energy stored in a different molecule called
ATP.
ATP. All right, so carbohydrates function as an energy source. Also, they're important in cell structures. They're a component of various parts of the cell as well. We often categorize carbohydrates into sugars and starches. We're going to get more specific. Like we said, the name itself, carbo meaning carbon, and then hydrate having to do with water. So carbohydrates are made of carbon, hydrogen, and oxygen only, and in a definite ratio. Right? And that there's a 2-to-1 ratio of hydrogen to oxygen. We generally recognize by their name compounds to be sugars with the OSE ending. Think glucose, sucrose, maltose, lactose, cellulose. All of these are sugars. There's some exceptions, but that OSE suffix generally indicates that we're referring to a carbohydrate. We will talk about first monosaccharides, the simplest of the carbohydrates, and then disaccharides, and then polysaccharides. Okay? And saccharide coming from Latin having to do with sugar. Like a sugar maple tree, its scientific name is Acer saccharum, and that saccharum comes from that same Latin meaning sugar. So monosaccharides. Mono means what? One. Okay. Monosaccharides, one sugar, that's what the word means, are sort of the building blocks, the units that are combined to form disaccharides and polysaccharides. Their molecular formula for the monosaccharides we're going to talk about is C6H12O6. And you will find that familiar. Why?
Glucose.
Because it is the molecular formula for glucose. But things get a little more complex because there are several types of monosaccharides that have the same molecular formula. And so what do we call, if they have the same molecular formula but they're actually not the same compound, what's the word for that? Yeah, they're isomers. So the three types of monosaccharides are all isomers. Okay? But they're all C6H12O6, but you could arrange those elements in a whole host of different configurations, and so they're each isomers. Okay? Now, just show you both of these. Love those. So we're going to talk about these three in a minute, but you can see they sometimes form open chains of atoms like this, but often they will form rings of atoms such as this. And so both forms are found, and they can be found in the same time, and they can readily change back and forth between the ring structure and the open structure. And so we're going to talk about three of these important monosaccharides. Okay. They're all isomers of each other. So first, glucose. So glucose is one we're probably most familiar with. It is the main source of energy in cells. The mitochondria uses glucose. It's an important part of cellular respiration. And many other types of sugars are broken down and transformed into glucose for use by the cell. Fructose is another type of monosaccharide. It's found in fruits, okay? Found in honey, in the nectar of plants and so forth. It's the sweetest of the monosaccharides as well. And then the third type is galactose. Okay? It's often a component of polysaccharides, can move through the cell membrane fairly easily. And so these are all isomers of each other. They're all C6H12O6. The empirical formula for all of them is CH2O. Okay, and that's one of the things, it's one of the ways you could recognize a carbohydrate is by that 2-to-1 ratio of hydrogen to oxygen. So if we look at these three monosaccharides, this is in their ring structure, which you will see them in most often. Okay, when you see that ring structure, that's going to be your signal that we're looking at a carbohydrate, at a monosaccharide. So fructose, glucose, and galactose. When we draw these in these structures, okay, at each angle of the shape is where there would be a carbon atom. Okay, so fructose is 1, 2, 3, 4, 5 carbon atoms. Okay, so it's a ring, it's a pentagon. Okay, versus glucose and galactose, which are what? Hexagons. There are a ring of 6 carbon atoms. Okay, and when you look at glucose and galactose, they look almost identical. Okay, so obviously you can tell fructose by it's a 5 carbon sugar, whereas glucose and galactose, what's the difference?
The H is
That hydroxide group. Okay, in that if you look here at glucose, this leftmost hydroxide group is pointing down, and in galactose it's pointing up. Okay? And that's the main difference. And the other thing that, as you get into advanced biochemistry, much beyond what we're going to do, there are also different configurations of glucose. In some configurations of glucose, this hydroxide is pointing up rather than down. And so it can get to be quite complex, but we are going to keep things fairly simple for our purposes. And again, you see here, this is the open chains of these sugars as well. And you can see, again, basically a repeating string of carbon atoms, okay, with the with some slight variations in the configuration, but they're isomers. Okay? So fructose is a structural isomer, which means its structure is different, whereas lactose and glucose are basically can be reversed. They're called stereoisomers. Okay, so that you see the difference is just in this carbon here is in the swap between the hydrogen and hydroxide. So a monosaccharide is one simple sugar. Disaccharide, di means two. So these are double sugars. These are sugars that are larger, that are synthesized by combining two of those monosaccharides. Okay, and so the formula for all of these disaccharides is C12, H22, O11. So again, you see there's still that 2-to-1 ratio that hasn't changed between hydrogen and oxygen. So, some examples, the three ones that you should be familiar with are maltose, lactose, and sucrose. Okay? Maltose. Maltose is found in germinating seeds. It's produced in the digestion of starch. You ever have malted milk balls?
Whoppers?
Yeah, like Whoppers, or a malted milkshake. Malted barley is used to produce beer. That's what malt is, or malt extract. So malt comes from that sugar. That's what gives that distinctive flavor. Okay, this is maltose. And what is it composed of? It's made of two molecules of water. So it's a disaccharide. It's two sugars combined. And include what sugars those might be? What's that? Yeah, it's two glucose molecules combined. They're combined here. Okay, if we look back over here at our glucose, okay, glucose where those hydroxides are on the left is down. Okay, combine two of those, okay, then you get maltose. We'll talk about how that happens in a minute. Another sugar, lactose, which you probably have heard of. Why have you heard of it? It's in milk, and so sometimes you may hear people are lactose intolerant. What does that mean? They generally will avoid dairy, and the reason is because they have lower levels of an enzyme, lactase, which breaks down lactose. So lactose is a sugar, okay? It's a sugar found in milk, and some people have, generally babies and infants have plenty of lactase because lactose is a sugar found in breast milk, and it's important for that digestion. But as people get older, some people lose the levels of lactase drops down, and they therefore cannot digest lactose or cannot digest it well, and it doesn't break down into its component sugars, and so it moves through the digestive tract, gets in the large intestine, water leaves the cells of the large intestine and goes into, because of osmosis, into that undigested lactose, leads to cramping and diarrhea, and it's generally not pleasant. The person that's lactose intolerant, what kind of things can they do to help with that?
I take lactose.
Yeah, and you know what, those lactate pills that you might take, you could drink milk that has that basic compound in it anyway. Basically, you're taking that lactase enzyme. And that lactase enzyme, as you take, helps to break down that lactose. Okay, so lactose is a sugar found in milk. Okay, and it is a combination of two sugars, again, put together. Okay, it's glucose and galactose. And then finally, probably the disaccharide you may be most familiar with is sucrose. This is just regular table sugar that you have at your house, that you buy, in a 5-pound bag at the grocery store. It's generally, it is produced from sugar cane, okay? And sucrose is another disaccharide. And so if we look at these, and here you have sucrose. If we look at these disaccharides, okay, actually we'll look at them in a minute, talk about how they're formed in a second. Then we have more complex carbohydrates. These are called the polysaccharides. What does poly mean? Many. It means many. And so these are large polymers, large molecules of repeating monosaccharides connected one after the other after the other. They could be 200 monosaccharides connected up to thousands of monosaccharides connected. And so if we were to draw a polysaccharide, we draw sort of like this. What does each hexagon indicate? A monosaccharide. And they're connected and they're bonded with an oxygen atom between them. So the three ones that you should be familiar with are starch, cellulose, and glycogen. Starch, we know, is found in plants. It's what plants, so we know plants produce glucose during what process?
Oh.
Photosynthesis. They produce glucose in photosynthesis. But then in order to store that glucose, they synthesize starch, which is a repeating molecule of glucose. Okay? And so that glucose gets stored in starch. It can be utilized when needed, taken out of storage, and then digested into glucose. Seeds have a lot of starch. In fact, when we get things like wheat flour, we are basically extracting the starch from seeds. That's what flour is. It's from the seeds. It's the starch from plants. And so why do seeds have starch in them? They need it. So starch contains stored glucose. Why is it found in seeds?
The seed can grow since it can't do photosynthesis.
Exactly. So when a seed begins to germinate, obviously it's a seed. It cannot go through photosynthesis. It has no shoots, no leaves. It can't go through photosynthesis to create its own glucose. So inside of a seed, most of the seed actually is starch. And it's some stored up energy, okay, that once the seed germinates, the plant starts digesting that starch, turning it back into glucose, which it can use to grow larger, eventually growing shoots and leaves, and then it can start to make its own glucose. But until that time, it needs to utilize that stored up energy that's in the starch of that plant. When you eat things like you eat beans, beans are seeds. And what you're eating when you eat a bean is the stored up starch that's in there that would have been used for that seed to germinate. Cellulose. Cellulose is another polysaccharide, and it's generally a structural polysaccharide. It's used to form the cell wall of plant cells. Okay? And cellulose is generally, it's not digestible to us. Okay? What kind of things can digest cellulose? Any idea?
Cows.
Cows can, right? Ruminants can, they're called. And so there are special enzymes that break down cellulose. And there's many types of bacteria that can produce this enzyme. In fact, in a cow's stomachs, they have symbiotic bacteria that live there and help them to break down the cellulose. It's difficult to digest.
Wait, what would happen if we ate grass?
We just wouldn't digest. So we do eat material with cellulose all the time. All vegetables have cellulose because they all have a cell wall, but we don't digest it. It's what we call fiber. It just goes through our digestive tract and eventually is eliminated in feces because it was not digestible. And then glycogen. Glycogen is another large polysaccharide. It's another molecule used to store energy. It's a polymer of glucose. And this is what animals use to store glucose, to store energy. So plants generally store energy in the form of starch. Glycogen is sometimes called animal starch. And glycogen stores energy. It's found there's a lot of glycogen in our liver to store glucose. Also, all of our muscles contain glycogen. So you probably remember when we have high blood sugar, what does our pancreas do? Produce insulin. What does insulin signal to our cells?
Lower blood sugar.
How? By taking in glucose. So it tells our cells, take in glucose because the level in our blood is too high. So an important organ in that is the liver, because the liver then takes in large amounts of glucose and stores it as glycogen. And that's sort of a reserve of energy that we have available to us. All of our muscles take in glucose and store it as glycogen. And so then if, for example, we stop eating, if we fast for a day, then so obviously we have no energy coming in. And so one of the first areas that our body turns to is breaking down that glycogen. That our liver will release glycogen, transform it, digest it into glucose, and that will provide the energy we need to live in the first day or so. Eventually that gets used up. If you ever run a marathon, okay, in a marathon you have to run for whatever, depends how fast you are, 2 or 3 or 4 hours or 5 hours. And in that process, okay, obviously you have some energy that you've been eating, you ate your pasta dinner the night before, but eventually running for that long you exhaust all of that energy you've consumed. And then your body starts to use energy from glycogen stored in your muscles. That runs out as well over the course of the marathon. And then you need to start, that's why people, if you run a marathon, you generally eat some sort of carbohydrates during your race, whether it's a protein bar or a gel or something. You consume those. What's that?
You run a pasta.
No, usually it's not pasta. Usually it's a little, you ever see the gel packets?
Oh, the Gatorade one?
They have Gator, they have all different brands, sometimes an energy bar, but you need to provide your body with some carbohydrates to keep supplying energy to those muscles. So those are the 3 types of polysaccharides. So how do these molecules get combined? Well, we're going to keep coming back to this process when we talk about each of these biomolecules. It's called dehydration synthesis. And you can imagine, I would guess, from the name what that means. What is synthesis?
2 things from
Building up a large molecule from smaller molecules. So this is how we take monosaccharides and combine them to disaccharides. How we take disaccharides, combine them to form polysaccharides. How we build large molecules from smaller molecules. What about dehydration? What's that? What? Yes, it's the removal of water in that process. Okay. So losing water and building molecules. So let's look here. What monosaccharide is this? What is it? Glucose. Look at this hydroxide group pointing down. What is this?
Glucose.
Also glucose. We can join these 2 glucose molecules together to form, you remember? Maltose. Okay? And the way that happens, if you look here, okay, at this first carbon here, and the 4th carbon here, okay, and you see they have those hydroxide groups, okay, and the hydroxide group here, and the hydrogen here, got H2O, okay, that water molecule is removed, okay, so we have water, taken hydrogen here, and an oxygen here, okay, and then the 2 molecules of glucose get bonded at this oxygen. Okay, so what you see here, once these 2 molecules have been combined, the carbons are linked through this oxygen. Okay? It's the remaining oxygen from here, and in the process, we lost a water molecule. And now these things are bonded together, okay? And so that's maltose. It happens through the help of enzymes, which we'll talk about when we talk about proteins. Enzymes facilitate a chemical reaction. They allow it to happen more quickly. They allow it to happen at a normal temperature. So enzymes help this process. They bring together these two glucose molecules, put them in a certain configuration so that those bonds are able to be formed. And this could be repeated many, many times, and it can form a polysaccharide. So this process is called dehydration synthesis. We're going to see it as we look at how proteins are formed and how fats are formed and lipids. They all happen through this process called dehydration synthesis. Okay? And so when we look at the overall formula for polysaccharide, okay, we have this sort of empirical formula. There's C6H10O5, okay? Times however many monosaccharides have been put together will tell you the formula for that polysaccharide. For example, when we talked about the disaccharides, what did we say their molecular formula was? C12. Yes. And so we have those monosaccharides being combined with the loss of water, and that should make sense right here, because when we combine two carbohydrates, we lose two hydrogen and we lose one oxygen. The opposite process is called hydrolysis. Hydro meaning water. Lysis meaning no. Lysosome breakdown. Okay? So this is water and breakdown. So dehydration synthesis, you remove water and put two things together. What do you think happens in hydrolysis? You add water and split things apart. It's just the reverse. Splitting a large molecule into smaller molecules by adding water. Do you remember what kind of enzymes we said were found in the lysosome? Digestive, but we gave them a name. We didn't talk much about it at that time. Certain type of enzyme. If you look at that word, it may.
What were they?
Hydrolytic enzymes. They are the enzymes that are responsible for hydrolysis. Because what does the lysosome do? Breaks things down. And it does that using these hydrolytic enzymes, these enzymes that facilitate hydrolysis. Okay? So, if we look and we take a disaccharide here, okay, sucrose, okay, sucrose is made of what? What is this? Oh, shoot. Don't look. What's that? Don't look.
Glucose.
What's that? Don't.
Fructose.
Fructose. Why you say what? Who knows and knows why? How did you identify this as something, Brandon? Why? Yeah, because it's that pentagon. There's 5 sugars. So yeah, glucose and fructose, they are what form sucrose, table sugar. Okay? And so if we take this disaccharide, 2 sugars, we add a water molecule and certain enzymes. Okay, you can see what happens to the water. Okay, an oxygen and a hydrogen are added here to this glucose, and the remaining hydrogen is added to the oxygen to form that hydroxide group. And now we're back to our 2 monosaccharides. We have a glucose molecule, we have a fructose molecule. So again, that's hydrolysis, adding water to split an atom. Okay, we can write it sort of in this way, showing a disaccharide being digested into 2 monosaccharides with the addition of water. we look at a couple other examples. Again, this is dehydration synthesis of sucrose. Again, it's made of a glucose molecule bonded to a fructose molecule. Again, 6-sided sugar, 5-sided sugar is fructose. Okay? We remove the water, we remove this hydroxide and this hydrogen, and they get bonded together at this oxygen. And now we have sucrose. Okay? We have that connection, and these are bonded together. And here we have the opposite, which we would call what? Hydrolysis. We're adding water to split this molecule of sucrose into glucose and fructose. Any questions about carbohydrates? All right, so that's carbohydrates for you.